And then Rob, you can we'll start right at 9. So we'll give
Sure.
it another minute for people to join in and then
Sounds good.
and you guys can still see the welcome screen, right?
I can, and I can see Eric too now. Yeah, I see Eric. Hi, Eric. Good to see you.
Perfect.
Hello.
Hello.
And when I'll move the slide over, Rob, when you introduce
Mhm.
Akeem. And then Akeem, you can just take over and I'll stop sharing.
Sounds good.
Perfect.
Maybe just one more minute and we'll start. Well, hello. Thanks everyone for joining today. I'm Rob Nickel. I'm the manager of the Bioindustrial Process Research Center here at Lambton College. We do a lot of diverse projects with a lot of very interesting company partners, and I'm very happy to introduce our company partner today, which is Canurta. We've been working with Canurta for over the past year on developing some new methods to look at cannabis phytochemistry. We have Akeem Gardner, the CEO of Canurta today, and we also have Eric, who is also a very accomplished phytochemist with experience in secondary plant metabolism and biochemistry, working at the University of Guelph. So very pleased to welcome both of these guys here today. And on the Lambton side, we've been working with Jessica and Steve Ganter in the lab and been utilizing all of our very nice high-tech chromatography with different types of detectors and all sorts of different methods to really take a deep dive into cannabis and to try and understand a bit more about the chemistry and the impacts on health. So without further ado, I'd like to welcome Akeem Gardner from Canurta. He'll give a talk about his company, where it's going, and Eric, I think, will join in as well and help out. And in terms of questions, if people listening today want to type questions into the chat, then I can facilitate some questions at the end of the talk today. So thanks very much for joining today, Akeem. This is our first Industry Insight. We used to have it under a new name, a different name. This is our first iteration of the event under the title Industry Insight. The goal is to talk with the private sector and try and learn new and exciting trends about what's going on in each of their respective industries. So today we're talking about cannabis and cannabis therapeutics. So thanks so much, Akeem. You're on mute now, but if you want to take it away, very happy to listen to what you have to say today. Thank you.
Everyone, it's a pleasure to be here. It's a pleasure to be here. I'm so sorry though, because of course, right when we're about to start the meeting, does my nephew come down and want to spend time with uncle? So if you hear kids in the background, please don't mind us this morning. Rob, I'm excited to be here. As you mentioned, we've been working together over the last year, specifically on this project. But as you know, our relationship goes back many of years. The cannabis and hemp industry has been an exciting one with a lot of research, development, a lot of trials and errors, and people who have persevered through with the right relationships, with the right partnerships, are getting ready to do some really, really exciting things. So today I'm excited to be here at your first Industry Insights meeting to share our story at Canurta, a little bit about what we're doing with botanical drugs. Eric will, or Dr. Soberanes will come in shortly in our presentation, and he will share a little bit about the specific project that we're doing with Lambton and how it's adding value and helping us on our journey to helping patients that need our help the most. Okay. Awesome. Now, I'm going to start, and I'm going to start with telling you guys a little bit of a story and a question that I have. I can't see anyone in the audience, so I don't know what we're going to say to this. But maybe Rob, maybe I'll ask you, what do you feel when you see this picture, when you see McDonald's?
I feel hungry. I haven't eaten anything today. I do love a good Big Mac and French fries, but
Yeah,
then
yeah,
I worry about, yeah, health impacts.
right?
Yeah.
It's very interesting how that happens. A lot of people, when I show them this slide, they'll say hunger. They'll say maybe a little bit of nostalgia. They'll think taking their kids to go get Happy Meals. The point of the matter is that McDonald's, as one of the most popular brands that we have, we can't escape it across North America. Did any of you guys know that there's 15,000 McDonald's we have across North America, 13,500 in the US alone? And again, you can't escape not thinking about Ronald McDonald, clowns, food, hunger. It's really a testament to the company and the brand's success. You would never think of McDonald's as being unpopular or rare, which is interesting because when it comes to diseases like ALS, amyotrophic lateral sclerosis, we have 2 to 3 people suffering from it for every McDonald's we have. Yet we characterize this disease as rare. Now, to everyone, to the general population, we may think of it as such, but to people who have it, it's anything but. Now, many of you may know ALS from Lou Gehrig's disease after it took down the famous Ironman after playing 17 great seasons, who rapidly deteriorated and passed away on the disease. Some of the rest of you may know it from the Ice Bucket Challenge that went popular 10 years ago. A lot of us saw famous influencers do it. Maybe we never understood why, but we participated. What many people don't know is that over the 150-year history of the disease has been 100% terminal. Everyone who's gotten the disease has, on average, 1,000 days, just over 3.5 years, slowly deteriorated, losing their ability to walk, talk, eventually breathe, and they lose their muscle mass in their chest, which leads to them suffocating on their own saliva at death. Imagine how horrible a disease like this is. What's worse is that 90% of the disease is sporadic, which means there's no rhyme or reason to how you get it. It can happen to any time, anyone, strike, and then you're on this 1,000-day journey. The other 10% of the disease is familial, which means if your family is part of a genetic carrier who has it, it can take loved one after loved one, family member after family member, generation after generation. We at our team, we met a lady, Erica, if you remember the lady we met who told us 24 people in her family had passed away from this horrific disease. Can you imagine what it's like to live life with that? Even worse, the disease is completely heterogeneous, which means it manifests differently in person to person. Person A's ALS is different than person B's ALS, which is different than person C's ALS, so on and so forth. This makes finding a solution extremely challenging and diagnosis even harder, which is even more worse because if you're of a minority community, if you're Black or Indigenous, you get diagnosed on average, uh, 8 months after our white counterparts. So imagine that. You have 1,000 days to live, 240 days not on any medicine, wondering what's going on, thinking that maybe are you making up stuff in your head or not? Who knows, right? You just have a bunch of wasted time. Now, with ALS, there are medicines on the market, but they all do not work. They're minimally effective, right? And the patients, their families, have expected for pharma to step up and to find a solution. But unfortunately, the way how pharma companies traditionally goes at solving diseases is they pick a target and they try to solve it. But in heterogeneous diseases, sporadic diseases, this does not work. You can't just pick one thing, solve for that one thing, and expect that you're going to solve the disease for the whole population. So now the key opinion leaders, the patients, and everyone involved in this industry are calling on scientists to think about things a little bit more broader. It's too complex a disease. We have to tackle it in multiple ways. And surely in nature, there are solutions, there are tips, right? Hints about what we can do to be able to solve such complex diseases. Now, this lady is Miss Kathy Jordan. She was diagnosed with ALS in 1986. She passed away earlier this year, rest in peace, after living 38 years with the disease. And Kathy is who's known as the patron saint of medical cannabis in Florida. She famously said she smoked the plant and she felt the disease stop. But it's not just Kathy, it's people like her who report in patient advocacy groups like Patient Like Me, like cannabis helps them with things like their pain, their sleep, their spasticity, a variety of their symptoms. It's not only patient reported, but it's also been studied in the literature. ALS patients use the plant, but why? Now, this is Dr. Ethan Russo. He's our senior medical advisor at Canurta. He's a board certified neurologist, and he's who we generously describe as the Michael Jordan of cannabis research. He's part of the team who brought the only two cannabis-derived medicines to FDA approval and global regulatory approval in Sativex and Epidiolex for GW Pharma. And Dr. Russo has a personal story with ALS. He actually has two. First, his wife's grandmother passed away from the disease, so he was always personally motivated to be able to solve for it. But on the second side, when Dr. Russo was a doctor back in 1993, he saw a gentleman, much like Kathy, who after being diagnosed with ALS only wanted to be treated with the plant. And over a 10-year period when he was under Dr. Russo's care, that patient never got weaker. So this led Dr. Russo to wanting to investigate, hey, what in the plant might be able to help these patients live longer, better lifestyles? And when he met us at Canurta as a Canadian-based company and saw the technology we were building on, he said he was going to help us understand how to develop the plant into botanical drugs. So a couple of the things that he taught us and a couple of the things that we're working on now. The first thing that you have to know when you're dealing with the plant and when you're dealing with botanicals is that many of these plants have long histories of traditional use, which are very important when it comes to understanding the proof of concept of disease area that they might be able to work in, but also their overall generally accepted safety profiles. If we look back in history, as far as 2700 BCE, over 5,000 years ago, Emperor Shang Yan wrote, protracted of taking of the plant may make one fat, strong, but never senile. It's very interesting to know because even if they had a little bit of munchies back then, they're going to stay strong and it's going to protect the brain. In India in 1200 CE, they wrote, in combination with celibacy and a strict ritualistic ritual, people who consume cannabis can live 300 years free from any disease and sign of old age. Again, I can't see any of us in the audience, but does anyone want to try? 300 years celibacy? Maybe? Who knows? In the 9th century, Al-Kindi in the Middle East and Syria wrote, hashish eases the muscles of the limbs and what flows. So again, they knew this was good for spasticity. They knew this was good for spastic diseases. Now, building on this, more recently in modern times, again, as I mentioned, the different ingredients in the plant have been developed pharmaceutically. People use them for multiple sclerosis. They use them for epilepsy, for Dravet syndrome, for early phases of childhood onset epilepsy. And researchers, when it comes to ALS, they have long called for how we can look at the plant and its different constituents to help with symptomatic relief, neuroprotection, slowing the disease progression. They even have calls for clinical trials, but still gaps exist. These are the things that we get to build on top of at Canurta today. One of the clinical trials that I'll point out is this study that was called the Canals trial, and it was done with Sativex, one of the pharmaceutically developed medicines that's used for treatment in multiple sclerosis today. In this Canals trial, they took 60 ALS patients, randomized them 1-to-1, gave them drug and placebo, and in the trial, on two outcome measures, the modified Ashworth scale for spasticity and the pain numeric rating scale, Was the trial statistically significant and successful in? Other secondaries like the ALS functional rating scale, spasms, sleep, and more, it didn't quite reach that threshold of statistical significance, which is great for us because now we have a proof of concept as to why our medicine ultimately might work. And we have an understanding of where the opportunities are where we can optimize our formulation further beyond Sativex to be able to help in this patient population. And that's exactly what we've done with our lead candidate, CNR 401. This is an innovative composition properly put together with the right amount of cannabinoids, terpenes, and for the first time ever, a prenylated flavonoid found in the plant called canflavin A. And as we go through our presentation, I'll leave it to Eric a little bit later to tell you a little bit about why we think this mixture is going to be helpful and some of the benefits of the canflavin A. Now, from an overall summary, why this is important for ALS patients, if you remember the other slide that I showed you, traditionally pharma companies want to pick one target and solve for that one target and help the patients, but it doesn't work in fast progressive terminal diseases like ALS. But with botanical medicines, with complex mixtures where you can have multiple ingredients in that mixture that do multiple things, you get to attack the motor neurons with multiple mechanisms of action. That means in a heterogeneous disease where person A's ALS looks different than person B's, both of them will get something that works for them after taking doses of our medicine, despite how different their disease may be. It's because of our multi-target benefits. Now, in order to prove this, we've had to do some preclinical trials. Of course, all this sounds good in concept. So here's some of the data and the results that we've done ourselves to be able to show how this works. I'm going to call out one experiment. And what we did is we used the zebrafish for our screen because zebrafish share a variety of genes with humans, about 70% of all genes they share, and 84% of diseased genes. So what that means is when you treat the zebrafish, you're able to get a pretty good read on if this is going to affect the motor function in humans. We used BMAA as a neurotoxin to simulate sporadic ALS. So what you can see is that with the normal zebrafish here, you give the BMAA, you reduce the distance moved, and then edaverone, one of the standard of care medicines that ALS patients are getting, was able to restore the motor function. And then we have 3 different versions of our medicines, with 401A being the ideal version that we aim to get into the clinic. And you can see this one works at least as good as edaverone, seems to work slightly a little bit better. And if you look at our videos, you can see this video kind of illustrates it. The no treatment arm, the BMA8 zebrafish moves half the distance in this experiment. You can see when you give edaverone, you rescue the movement, right? But then you can see what it looks like when the zebrafish larvae are on our drug versus edaverone. It seems that they have just a little bit more zest. Right? Now, after that experiment was done, we sent the zebrafish to the NRC. We extracted the RNA from the zebrafish, and what we did is we did some RNA sequencing to understand the differentially expressed genes and the therapeutic potential of our ingredient versus edaverone. And the big takeaway here is that when you look at our CNR 401, sorry, we differentially expressed over 900 different genes compared to 480 with the edaverone. Our asset just seems to have a much broader therapeutic potential than the medicines that the patients are getting today. In the upcoming months to years, our job is to narrow down these different genes and see if there are things that we can follow that could be promising biomarkers in humans when it comes to clinical trials, and how this is going to help us understand not only how we can help with ALS, but other neurodegenerative diseases like Alzheimer's dementia, diseases that have neuroinflammatory biomarkers, so on and so forth as we go forward from here. We do this because, again, when it comes to the competitive landscape, I've already explained the standard of care medicines. They minimally work for neuroprotection. They provide no symptomatic relief. All of the medicines that patients have to take carry a host of adverse events. This is just where we have the ability to win and provide patients something that can be the new standard of care, helping slow disease progression potentially, helping with their symptom management, improving their quality of life in a minimally invasive way. So this is what we're working on. We will do this, and we have different multi-target solutions that help us do this. Eric is going to talk to you a little bit about, well, before Eric talks to you about how we standardize our medicines, what the CMC required for botanical drugs, the one thing that I will say is that we have a suite of patents that allow us to produce these ingredients. We can extract them, biosynthesize them, and we're even working on growing these molecules in the future. And we start with ALS because this is the disease that we have the most evidence for, plus it has the most urgent need when it comes to the patient population. But this asset has pipeline and a product potential, which means it might be able to help with other dementias, other neurodegenerative diseases like spinal muscular atrophy. And then from here, we'll also be able to help in other inflammatory related diseases like rheumatoid arthritis, osteoarthritis, IBD, apoptotic dermatitis, so on and so forth. Now, before I pass it to Eric, one of the things that a lot of people might say is that when you're dealing with botanical therapies, how do you protect them? Because you can't really patent the plants. So what you have to have is the ability to think around that and understand with foresight, you have to patent not only your production methods, but also your therapeutic compositions and the use of those compositions in those therapeutic areas. We've also started to patent different ways that we can use AI and machine learning to optimize our medicines, our formulations, our NCEs. And you can see that we've planted our patents strategically around the world in major markets where we think that we can have commercial success. But the other way of getting defensibility is this area, the rigorous CMC required for botanical therapies. What many people don't know is that the FDA and the World Health Organization, the EMA, they all have standards and guidelines that you could go down to be able to make plants not only natural medicines and supplements, but also botanical drugs. And when you can get your CMC up to that standard, this is an extra layer of defensibility on top of your IP portfolio. Now, this is the basis of the work that we're doing with Lambton College and why it's so important. So now I'm going to pass it over to Eric to explain a little bit about what he's been working on when it comes to botanical extracts, drugs, and what's going on here with CNR 401. Eric, over to you.
Thank you so much, Akeem. So yeah, I'm going to talk a little bit more about the Canurta botanical extract, or CNR 401. So Canurta botanical extract is hemp-derived, of course, and it's featuring proprietary blends of cannabinoids, terpenoids, and canfavin, all from hemp. It's of course, the production, the manufacturer needs to follow standard regulation, as Akeem mentioned. So it requires botanical drug to maintain consistent quality and composition across batches of the botanical raw material and the drug substance and drug products. So that's what all the FDA, EMA, and even Health Canada will ask us, in fact, to go to clinical trial. So, in fact, the term of chemical manufacturing and control, so CMC, covers the various procedures used to assess the physical and chemical characteristic of the drug products and to ensure the quality and the consistency during manufacturing. Here, I also added a few of the compounds that we have in our CNR 401. So, of course, Canurta developed an extraction method from hemp biomass waste to produce its pharmaceutical. We wanted to be able to reuse biomass that have been already used to produce hemp oil in the regular standard process to be able to extract our polyphenol extracts. So it's a little bit of sustainability on that side. Of course, our extract is a mix between hemp full spectrum extracts. There is also polyphenol extracts and more compound like terpene and other cannabinoids that we also have to add later on. it's a complex manufacturing process. We have partnered with multiple entities, mostly InnoFiber in Quebec, that is also a college, applied college, and they are also helping us on a daily basis. They have done tremendous progress on scaling up the production, in fact, of the extracts, of the polyphenol extract mainly. And we have a partner in the US called FS Oil that is helping us producing and manufacturing all the extract all together, and will also apply the polyphenol extraction. So how do we ensure that all this manufacturing process is comply with regulation? So the technology process is currently being improved, and as I say, we have to be careful of the complexity of the extract. Here on this graph, it's an example of Sativex. So Sativex is one of the botanical drugs that is already on the market, and it's a composition of the drugs that you have. So they have to be aware of all the components of the drug. As you can see, Sativex is mainly composed of cannabinoids. It will be THC and CBD, but you will also find other like triglyceride, terpene, trace of ethanol, fatty acid, and also unknown compound. So the issue with botanical extracts that you have numerous bioactive compounds. It requires batch consistency to maintain its chemical profile, and it's critical to preserve efficacy and quality. So to meet this standard, Canurta has developed its process identifying different principal active components, and those principal active components will be key indicator of the consistency and the potency between batches. So by standardizing the component, Canurta guarantees that this bioactive property of the botanical drug, in fact, remains stable and effective throughout each production cycle. So now, how do we control the botanical product? There is multiple rules that we have to apply, and we have to follow step by step. We have to develop test method and limit to ensure that we have identity, strength, quality, and purity of CNR 401. We also have to do a description of analytical test methods, LCMS and GCMS, and this is where Lambdon is helping us a lot, because we have sent them our pharmaceutical CNR 401, and they are helping us developing analytical methods specific to our pharmaceutical to detect the different active pharmaceutical compounds that we need. So of course, we're going to have to propose limits based on the analytical development and batch data to report to the regulatory entities. There will be specification tests that will be applied with the different testing, the method applied, and all the acceptance criteria will be developed following those tests. And accumulating different batches will help us standardize our extracts and also showing stability of our extract to the different regulatory agencies. So we also need, as you remember, I mentioned that botanical extracts are complex mixture. So we need to characterize all the constituents present in the drug product. Why? It's just for safety. If you are going to the FDA and you show them that you have botanical drugs, they want to know what you have inside. And they want to make sure that's going to be safe, so you don't have anything toxic. And it's also stable and consistent, that every single batch will be the same. That's why you need a complete characterization of everything. So this is when I'm going to introduce the term of chromatographic fingerprinting. So it's pretty much using the different methodologies that we developed with Lambdon College. We're going to be able to have a complete scan for different major class of compound like cannabinoids or terpene or polyphenol. And we'll be able to identify each single species. By identifying each single species, we'll be able to have a full characterization of the extract. The fingerprinting, in fact, will be the following step. And the core of the fingerprinting methodology, it's a technique that generates a detailed profile, in fact, of the chemical composition of the botanical extracts. The profiles that we obtain with the mass spectrometry serve to describe the unique chemical characteristic of each batch, and a reference standard will be established with different samples, as I mentioned before. And this will generate, in fact, the fingerprints of the extract. And afterwards will be all the subsequent batches are analyzed will be analyzed by a third-party analytical laboratory using the same chromatographic method, and the potency and the fingerprinting will be compared. Any deviation, in fact, from the reference profile will be flagged, and we will we will see if you have any potential inconsistencies. So this is how it's working, and this is why we have developed a partnership, in fact, with London College. This is mostly to apply all those regulations and develop all the CMC regulations that will help us to go through preclinical trial later on. And I think that's it on my side.
Thank you, Eric. Now, as you can see, all the work that it takes to get a botanical drug in order, not only on the therapeutic and the efficacy side, but on the standardization side of that drug, is extremely tedious. There's a bunch of things that need to be done, and all of it in preparation for your phase 2 clinical trial. But luckily for us, we have a great team and we have good partnerships that are allowing us to get to the point where we're almost ready. We're looking for in the first half of 2025 to initiate a trial called the HALOS trial, which will be our in-human phase 2 clinical trial with CNR 401 in ALS patients. Now, I won't go into this too much for today, but if anyone has any questions, I can describe how we think we're going to help. But long story short, this will be the first step on a long journey for us that can help patients improve their symptoms, their quality of life, and see if we have disease-modifying potential as well. As you've seen, in companies like ours, and in any company for that matter that's trying to do something so big as to affect a disease for a patient population that has very limited to no answers, it takes a village to be able to get there. I can talk for days about the team that we've put together with our leadership team, the all 10 of us that are responsible for building the cell, or our world-class science advisory team and our industry partners who have all contributed in multiple ways in this journey. But the one thing that I did want to leave you with is that when it comes to entrepreneurism, or when it comes to what any of you might be looking to do when you graduate, it's very important that you understand the why of what you're doing, because this is what helps you persevere when things get tough and when things get challenging, and everyone needs a reason to enjoy wanting to go to work, not just going to work because it's what you have to do. These are my grandparents. My grandparents came to Canada from Jamaica in 1972. They brought my mom here with them. They had my aunts and uncles. I was born here in Oshawa, so I'm the first grandchild of the bunch. I'm not saying I'm the favorite, but I am the first grandchild of the bunch. I grew up playing basketball, so a lot of the things that I learned come from my experience in the world of sports. I've always thought that you need a team to win a championship. So again, in our presentation, you heard us giving credit to all of our wonderful partners that add to our mission of winning our championship ourselves. After that, when I grew up, I played some basketball at the University of Ottawa. I got my degree in psychology and philosophy there, after which I coached for 2 years before I thought I wanted to be a general manager of the team, kind of like Masai, putting the team together to win a championship. So I went off to law school, and when I came back from law school, this is where things changed for me. Back in 2017, when Canada was legalizing cannabis, I knew that there was an opportunity for this plant to be a meaningful part of the solutions for our future, not only in regards to climate resiliency and providing foods, but also medicines that you can get from the plant. The second thing that happened at that time, though, and little would I know how this would personally affect me, but interesting today, is that my grandfather was diagnosed with Parkinson's. this is my grandfather today. This was very meaningful because when he was diagnosed, when I was looking on YouTube, I kept on seeing videos of people using cannabis oil or hashish and helping with their muscles, their tremors, just like how Al-Kindi said in the 9th century. And this made me really, really excited to know or to get ahead of all the opportunities that Canada was giving us because we legalized first before the rest of the world. Eventually, everyone else was going to catch up. But by 2030, if we paid attention to the plants, if we dug deep enough, we would get the ability to understand how really this could be used to help the next generation of human and animal health. So what did I do? I did what any good entrepreneur does. I had a line of credit that I used to go to law school. I took it and I rented some farmland north of Orangeville, and I started my own farm. No equipment, just my hands, my feet, some craziness. Hey, friends, family, come help me harvest the hemp. This is how we did the first 30 acres that first year. And this, though, was the scariest decision that I ever could have made because, again, who goes to law school and then ends up on the farm? Watching all my Canadian classmates become lawyers and look at me there with no clue what I'm going to do with all this hemp. But what I was able to do is I was able to invite people from University of Guelph, Lambton College. I think this first year, Rob, if you remember, we started a project where I gave you our hemp fiber and we were trying to investigate 3D printing materials for building materials. We just tried a whole bunch of things. Little did I know that this decision would have been the best decision I could have made because it put me on the same level as all the farmers in Ontario. It allowed me to start a wide variety of research projects and relationships. And now, fast forward, we're studying ALS. We have a medicine that can help here, but ALS also exists on the spectrum of other neurodegenerative diseases like Parkinson's and Alzheimer's as well. Now coming back around, who knows what our science and our investigation is going to lead to be able to maybe help people that are like my grandfather in future years. So again, I ask you, when you're driving around and you see McDonald's from here on out, don't just get hungry, don't just want to go get your Happy Meal, but think about the people who need our help, the 2 to 3 people who are suffering from this traumatic, rapidly degenerative motor neuron disease that are all fighting for a better tomorrow. Think about their friends and family who are personally advocating for change, who need our help, who are advocating for governments to help, and pharma companies to step up. And remember that magic can happen even here in Canada when you build as a team. We're doing this work and we're doing it right across Ontario, not only us in this picture, but with our partners as Lambton, because we know that in nature, in the power of plants, there are solutions that can help people tomorrow. And we're starting with ALS because ALS is not rare. It can happen to anyone. And these patients need our help the most. So that's a little bit about our story and how we're going about things as Canurta. I will ask now, do we have any questions about anything that we're doing? I'm here to support.
Great.
Let me come back and come over to you guys so I can see everyone.
Great. Thanks so much. Thanks so much, Akeem. Yeah, on one of your last points there, I find it interesting that, or hopeful, I should say, that Canada was one of the first countries to legalize cannabis nationally at the federal level. So I see that as a natural head start for Canada, and hopefully we can grow some of these therapeutics, discover, grow, and help with these target populations that definitely need help. So I think Canada is a great place to be in terms of this types of research and development, and I think Canurta is definitely ahead of the curve and really making great strides towards those goals. So thanks so much. We're getting virtual applause, I think, and for your talk. It was very nice to hear and great to hear what you guys have been up to. If anyone has questions, please just type it into the chat. I have a few already. The one I had off the bat was, you mentioned Health Canada and the FDA. Are there any learnings there about, the different regulatory between the two countries? Is one easy, in terms of a young entrepreneur or, a startup pharma company, is one route better than the other? Or are there some kind of regulatory considerations for the two countries that differ?
Are we talking about in regards to and it's because it's a loaded question. Are we talking
Right.
about in regards to getting drugs? Are we talking about the difference between drugs and supplements between countries? Or is it
Oh,
just
I was
research in
thinking
general?
drug approval between
Mhm.
the two countries.
Well, again, Canada, we are in a very lucky spot to be Canadian
Mhm.
when we're doing this. And back in 2005, Health Canada was actually the first country to approve Sativex, a one-to-one CBD to THC medicine for multiple sclerosis, before the rest of the world. So again, our country has a history of being open-minded, of being able to get medicines to patients that can help, and thinking about botanical drugs, cannabis-derived medicines, but other plants in a very, very innovative way. So one of the key things that I would always suggest is engagement with the regulators. Even though the FDA has a well-defined botanical drug guidance, you can Google it. Health Canada doesn't have one. If you go talk to Health Canada, you can pretty much get the feedback and the opinions of what you need to be able to move things forward. We also are very lucky because we have a lot of consultants and a lot of people who can help. And even though some people may say that Canada, we're a small country in our population, sometimes there's power in that small population versus in the States when you're one of across how much? 400 to 500 million people. In Canada, we're one of 40. So it's easy to get in touch. It's easier to get in touch with our regulators, with our members of parliaments, with the patient community groups that will advocate on your behalf when you have something that you believe can help and you have rationale for why it can help. So always keep these things in mind and go talk to the regulators because they're there to support and help you.
Very good. How about that Halo trial, that clinical two? By the name, I assumed it was maybe several different products, maybe different companies or different pharmaceutical products, or is it basically mostly on yours? It's just your Canurta CNR 401 will go into clinical two?
Yep. The Halos trial. Halos is an abbreviation, and it sucks because I can't remember it right now. But it's our first in human clinical trial for CNR 401 that will go into patients. Now, the interesting thing about Halos is that even though it is one drug, when it comes to clinical trials, especially in rare disease populations, you have to have global participation to be able to recruit the patients required for this trial. So our trial will actually start in Washington State in the United States. Then we'll bring it to Canada, we'll bring it to the United Kingdom, and then we'll go to Australia as well, targeting 50 patients in each geographic location. That will get us our 200 patients that we need to get enough power in our trial to be able to then go back to regulators and say, hey, look at our effect size. We think this works. How do we get this on the market as an approved drug with an approved claim for the much broader patient population?
Great. That sounds exciting and a great segue to this next question. So, as a clinician, is there anywhere I can refer a patient to get involved in a clinical trial? So, I guess not the Washington one, but perhaps the Canadian one coming up
Yep.
or planned.
So I would 100% start with talking to the people at ALS Canada and the
Okay.
Canadian ALS Network, or CALS in short. CALS is all the Canadian neuroscientists and clinicians that are open to clinical trials, that have active and ongoing clinical trials, and they are the ones who will take on our trial when we're ready to get going in 2025. The nice thing about the ALS network and the nice thing about CALS is that they make really, really good relationships with our academic and our hospital institutions. They work well to screen rapidly and fastly the safe and promising therapeutics that have the potential to help. And they are the ones who will be able to tell the patients exactly what they need to know when going into the trials, collect the informed consents, so on and so forth with the work that we're trying to do today. So I would always start, if it's ALS specifically, ALS Canada or the Canadian ALS Network. But despite what I'm doing specifically, ALS is just our where we're starting.
Mhm.
Every other disease has their community advocacy group. You have Arthritis Canada, you have Brain Tumor Canada, Brain Tumor Canada, we have Cancer Canada, we have Rare Pediatric Disease Canada. So if you work in therapeutics and you have an idea of a plant, a synthetic medicine, a protein, or whatever it is that you think can help, I would always start with taking whatever promising data that you have and going to the community group that you're looking to serve and say, hey, I'm here to serve. I don't really know how, but look at the science, look at the data. Can you point me in the right direction? This is how we started with ALS. And then a year into our process, now we're way more confident about what we think we can do because it's those people, it's those scientists that helped us fill the gaps into where the need was. And then when we go back to our partners and we see what we can do on the bench scale, it just makes us excited for what we think we can do going forward from here.
Great. That's great advice. That's a great answer, actually. Very informative. So thanks for that. I had one about, I'm not sure if you can tell me, some things proprietary, et cetera, but the gene expression, the RNA type of studies. Can you tell us, are there any clues about, mode of action at the genetic level? Anything happening there? I think you mentioned 500 genes kind of stuck out. Anything that you could tell us about that?
Oh, these are very, very exciting. And I'm going to answer that question in two ways because the first way is, and all of you heard a little bit about my background. I'm not the lead scientist, so we're still learning. And funny enough, even though we have scientists on our team, we have our bioinformaticians on our team, we are all still learning about what the real potential of what we can do is. Now, one of the things that I'll show you, and I'll just highlight it on this page because it's easy to share, is that after we did our gene sequencing, sorry, I tried to zoom in, but maybe you guys can see here.
No, I didn't. It worked. Yeah.
Yep. What you can see here is that what we were able to do is we were able to group the gene ontology terms to see what groups of genes we have really good activity in. And unique from edaravone, one of the standard of care medicines, you can see the one that pops out are gene ontology terms that have to do with the anti-inflammatory response, which is fitting because our prenylated flavonoid canflavin A has 30 times the anti-inflammatory activity than ASA, the active ingredient in aspirin. Plus, we know that other cannabinoids and terpenes in the plant are anti-inflammatory and are neuroprotectants as well. So right now, what we're doing is we're screening through all of these and saying, okay, we understand the groups of the gene ontologies, but are there specific biomarkers that translate really, really well from animal to human studies? When we look in human databases like the UK Gene Biobank or the PROACT, which is an ALS data center that takes post-mortem ALS patients and natural history studies where they give biomarkers in the duration of their disease. Are we seeing upregulation or downregulation in specific genes? And is our preclinical results matching? I explain this like this because, again, depending on who the audience is, the point that I'm trying to make is that in research and development in science, there's a breadth of expertise that's required to be able to bring drugs to market. It's not only the plant scientists, it's not only the people running the HPLCs, but it's also the bioinformaticians, the people that are good with data science, data science, bioinformaticians. It's the people who can just ask the questions. Again, I come from the legal background, but I'm very good at asking questions and trying to use my deductive reasoning to help people like Eric and the rest of our team get us to the answers of what we need. There's a whole bunch of research that needs to go on. And I heard this on the weekend at the ALS conference. Research means research, which means you search again, you search again, you search again, and you keep going until you get the answers that you're looking for. So it's really a multidisciplinary approach that's going to lead to success. And that's the point that I'm trying to drive home here.
Okay, thank you. And I mean, the chemistry, of course, we know is complex. I don't want to add more chores to our list, but just the thought occurred to me. Well, you as well. I know. AI might be a great tool for all these molecules, all of these genes, all these pathways, all these outcomes. Yeah, and to have pathways not engaged by that standard of care, current standard of care, have your product engage those pathways and genes is very exciting, but a big task to sort everything out. So maybe a question for AI and machine learning too.
Exactly.
Thanks for that.
The one
I
thing
think
that I'll say
Mhm.
I'll say here just to that point is that like any team, and again, I use a lot of team sports references. So for
Mhm.
any athletes in the audience, you'll all understand this. But everyone understands this because organizations, teams, we always work better when everyone's moving in the same direction towards a common goal, but everyone is also allowed to play up to their strengths. So you don't have to be an expert in everything, but what you do have to have is people around you that you trust, that you can rely on, that you know who are just as good as at their job as you are as good as yours. I played on a lot of basketball teams my whole life, really, really talented ones. For anyone who knows the Canadian basketball ecosystem, you know that we've had some of our Canadian talent make to the NBA. Some of these teams that I played on had some of this talent, but we never won championships because ego or people didn't buy in, so on and so forth. But then there are teams that you play on where everyone knows their role, everyone knows what they add to the overall mission, and everyone trusts each other. And usually, when you can find organizations that work in that way, that's when you can make magic happen on and off the court. So I always try to think about that in regards to not only what are the skills that I need to develop, do I need to become a machine learning expert, et cetera, et cetera, or can I find us the right partner who has the infrastructure who can help us do what we need to do? And again, this is where knowing you, Rob, and Lambton College, as soon as we had this project and we knew, hey, you guys can help us characterize the polyphenols, Eric, we have the right partner for us to be able to amplify our work and help us get to our ultimate goal, which ultimately helps us win the championship and helping these patients that we aim to serve.
Thanks so much, Akeem. That's nice to hear, and happy to be part of the team. Happy to keep growing with you, and a lot of great challenges ahead. Absolutely, that I think I agree we can all work together and maybe help. Of course, Canurta, but just back to that, the RNA. Who knows what kind of other impacts and other new potential pharmaceutical mode of action we can discover. So very excited. And it's been a pleasure to work with you, and we'll continue to work and keep everything rolling along here. So thanks so much. I don't have any other questions. Um, just like to thank again Akeem and Eric today for giving their time. We're going to wind down things for the holidays, but we'll pick things up in the new year and keep the chromatography humming along. And thanks so much for joining. Thanks to all of us, all the audience today for joining today as well. So have a
Awesome.
nice rest of your day. And take care, everyone. Thank you.
Thank you, everyone. Oh, and just so everyone knows, feel free to reach out on LinkedIn. Feel free to email me if you have any questions, comments. Eric and I are both available. Okay. Thank you all so much.
Thank you.
Thank you.
Take care.